Thank you, everyone. Good morning, good afternoon, good evening to some of you. I'm going to be giving really an overview of the past in connectomics, how we got from humble beginnings to where we are now. I'll spend 5 minutes, 3 minutes on the present, and then talk for the remainder of the time about the future. And yeah, I'm actually really glad that Terry had his first slide going, his classic slide, going from molecules to whole brains. And that's what I'll be talking about, particularly in the second half of the talk. What can we do in the whole brain now? And there's a lot of science going on in this space that probably most excitingly, immediately, right now, there are brain-wide surveys of cell types as classified with their transcriptome. And these surveys are well underway in the mouse and will soon exist for the human brain as well. So cell types classified by the genes that the cells expressed, with a nice amount of morphological, in some cases, even biophysical work. So this really is from molecules to mind, leaving out a lot of the details, but spans all the scales that Terry was talking about. In the human, it obviously would be great to measure the activity of neurons all over the brain, but this is currently impossible. There are better and better approximations of this in the mouse, but it's really a long way off. The million neurons, the 10 million neurons at once that people dream about, recording it throughout a mouse brain. But I want to argue that it is possible to make brain-wide wiring diagrams, connectomes. And for this, it's important to go back to, especially for a diverse crowd, some of whom might not have been following connectomics, to the beginning, with the definition of a connectome from Hageman and Sporns, a comprehensive structural description of the network elements and connections forming the brain, or even the human brain. And it's very important to, sorry, I'm moving my Zoom widgets around so I can see the slide. It's very important to emphasize there are two types of connectomics. The type that those of us in electron microscopy call connectomics, which is the level of single neurons and synapses, but there's also the level that MRI people who are doing diffusion imaging of axon tracts call connectomics, and that's at the level of anatomically distinct brain regions and interregional pathways. So again, I'll be talking about both of these, a little bit about EM connectomics. You'll hear a lot more from Nuno da Costa in the next talk, and about new ways of doing cellular resolution, macroscale connectomics. And I'm sure everyone, all 200 and whatever it is now, 92 of you have heard many talks or seen many slides that make the analogy between a computer chip and a brain, but I want to give a slightly new version that I haven't really seen before that makes the very clear distinction between microscale and macroscale connectomics, both for brains and for chips. So on the left, we have one of the biggest, if not the biggest, CPU currently available. I'll talk about it in a few minutes. And then we have the human brain. And I'm sure most, if not all of you, have seen images like this, which are from MRI and are sort of the emblematic pictures that one sees for human brain connectome talks and research programs. However, what I want to get at, this is not really a wiring diagram. The resolution is too low. I mean, it's a fantastic research tool that gives sort of the bulk directionality of fiber pathways at each pixel, but the pixels are each a cubic millimeter, so it's bulk, not at the neuronal scale. And when I was thinking about the problem of CPUs versus brains anew, I came across this video, which really, for me, opened up the thought of, let's look at a modern computer chip and slightly revise at least some of our views of what goes on in a chip. And if you're going to reverse engineer a chip, the only tool of choice is the electron microscope. And as you know, as an EM connectomicist, if you're going to reverse engineer the brain, you also need an electron microscope. But what do you see when you look at a chip with a scanning EM? And this is a video I found on YouTube. The reference is in the lower right-hand corner of what you see when you look at the top of a modern chip with a scanning EM. You don't see silicon. You see something that looks like city blocks, Terry just said, or a grid of wires. And these are actually fairly macroscopic wires that are at the surface of a chip. But the wires are roughly a micron now. They're a good deal smaller than that. But if you want to look at the scale of individual silicon elements at this scale, the wires are much, much larger than the silicon elements. And you can see this better in cross-section. If you can see this better, in cross-section, a slightly more modern chip that has 7 metallic layers going from top to bottom that are sort of coarse scale wiring between modules of a chip down to the very fine scale interconnects with the silicon itself. Modern chips have 10, 12, 13 layers of wires. And of course, these for me are the analog of the long distance axons, elements that don't in themselves compute, but connect computing elements. This is another movie I found 8 years old of how these are built up. So this is a logic gate. This is a movie from Intel. This is a logic gate and maybe an interconnect on top of it. But a chip is built by making an infinitude of logic gates, but then building layer upon layer of these passive these wires that make the thing compute by linking together billions of transistors to do computation. So to continue the metaphor, sorry, I'm getting rid of the little icons from Zoom. If you're going to understand a chip, you understand the silicon, of course, in the case of the biggest chip around, the AMD EPIC Roma, with 40 billion transistors. So you look at the 40 billion gates, but you also need to look at the macroscale connectome, the long distance wires. And of course, the analog holes, you have neurons, which are radically more complex than a gate. Maybe a synapse is a gate rather than a neuron. You have the long distance wires, and then you have the whole brain. And again, a neuron is far more complex than a gate, but the EPIC Roma chip has half the number of transistors as we have neurons in our heads. So part one of the talk is microscale connectome, the silicon. And I'll be talking about the cubic millimeter that Sebastian Sung, that we collected the EM data for. Sebastian Sung has finished the segmentation, and Nuna will talk about it a bit, but it's 100,000 neurons, as Andreas said, and 1 billion connections. And I found this about a year ago. In fact, I found this about 40 years ago when I was an undergrad. And for people of of a certain age, this is, for a large number of people in neuroscience, this is the first introduction to neuroscience, a little bit more than 40 years old, a copy of Scientific American. And I really urge anyone who hasn't seen this, it's a book as well, to look at it. Wonderful article by Hubel and Wiesel, wonderful article by, among others, Wally Nata about anatomy. And it finished with, of course, Francis Crick thinking about computations and the brain. And it was a wonderful paper by Francis Crick, and mostly about computation. And in it, he says, why do we need to make abstract models or reduced models? Because there are certain things we're never going to do. And I read this 38 years ago, but didn't remember it. He said, it is no use asking for the impossible, such as, say, the exact wiring diagram for a cubic millimeter of brain tissue and the way all of its neurons are firing. And it's a case of, for 40 years, all of us running a race that we forgot we were running. And this is getting too overblown, but it's, this is in a way a victory lap, that with the Baylor and Allen and Princeton teams, we've actually done this. And Nuna will talk about it. Andreas showed the 70,000 neurons firing. Let me do a quick survey of our life in connectomics, but really the way things have advanced so amazingly, excitingly in the past 15 years. The first couple of papers from our group and Winfried Denk in 2011, we were happy with a couple hundred or 100 neurons, a couple hundred connections, looking at inhibitory circuits in the cortex along with function. 5 years later, we had a bit more function, a bit more calcium imaging, that work done at Harvard with Wei-Chung Lee. But we were happy then with 1,000 connections of a couple of hundred neurons. And with that kind of functional connectomics, we were able to address a longstanding question that others had beaten us to the punch, but that neurons with the same orientation tuning, with the same function, tended to connect to each other more than neurons with opposite orientation tuning and encoded by color. But where are we now? This is very sparse tracing before machine learning segmentation happened. Now with the IARPA microns program, which I'll talk about in the next slide, we have dense segmentation of the cubic millimeter, 1 billion connections, 100,000 neurons. It needs proofreading, there's work to be done for sure, but at least in outline, it's all there. And this is kind of fun that IARPA had 2 programs, both essentially for reverse engineering. The program that allowed our distributed group to do this impossible cubic millimeter with calcium imaging, then EM reconstruction of the same neurons, and then using it as Andreas said, to help recursively machine learning to do the segmentation, and then the knowledge this all gets us to think of new primitives for machine learning. A year later, I won't talk about it at all, there's the Raven program that uses pretty much exactly the same tools to reverse engineer chips, again, funded by ARPA, same 5-year plan, and using roughly the same amount of imaging, in this case, SEM, to reverse engineer a cubic centimeter or a square centimeter of chip through the multiple metal and then silicon layers. The ARPA team, Nuna has this slide, so he'll show it as well. And Andreas had a slide like this. And very quickly, because I have to get to part 2, we did 3 volumes, a modest volume, which is huge by old standards, of 500 neurons, 3 million synapses, which is online now, which is one reason to talk about it. Nunu will tell you about that. Then something that allowed us to get used to doing 1 square millimeter sections, but this is a thin pancake of 1 square millimeter by 50 microns. And then what we've talked about, both me and Andreas and Nunu, about the cubic millimeter. And this is my last EM slide. Really, this is, Nunu will be talking about this in great detail. The wonderful segmentations, even from the small data set, that allow you to differentiate the different cell types. Here's a nice basket cell with 4,700 incoming synapses and 1,500 outgoing synapses. The numbers are greater for the large data set. And we're really going from molecules, transcriptomics, which Nunu will talk about how we can map transcriptomics onto morphology and morphology onto connectomics. And for people in the modeling world, another way to get at cell types is from just from the connectome itself. And I urge you to read this paper by Jonas and Cordy, 2015, with keywords, stochastic block models. If you have a connectome where the different cell types connect in their idiosyncratic way to each other, you can infer the cell types just from the connectome itself. Part 2, sip of coffee. A macroscale connectome. So I said I'd give you a few minutes of the today, and now I'll talk about the future, about macroscale connectomics. Something that I've been wanting to do for a number of years, and we finally have a team together and some funding to do it, which is to make a macroscale connectome, ultimately, of the human brain. And I prepared you for the computer analogy, but in the human brain, there are more than 10 billion long-distance axons. Together, they have 100,000 kilometers of total length. What is this wiring diagram, and how could you possibly get all of these wires, the actual data for all these wires? I'll rush through this again. Francis Crick was there thinking about this a long time ago. And this is a wonderful article by Crick and Jones in Nature, asking the question, what does a human connectional map look like? And saying, this is something we need. But the shameful answer is that we don't have any detailed maps compared to what we had even then in the primates, non-human primates, for instance, because for obvious reasons, most experimental methods used on the macaque brain cannot be used on human. There really hadn't been any new methods for long distance track tracing between when this started in the 1910s and quite recently. So this is what I want to discuss in the last few slides. So where are we now with long distance connectomes of the human brain? Again, with diffusion imaging, human MRI connectomics, we have these bulk maps of the bulk direction of axons. But if you, and you get this by stringing together vectors from these 1 millimeter voxels. But when you want to zoom in on this, all you have is essentially the data. And in fact, these wires are interpolations from very coarse data. These pseudo wires are actually at a far finer scale than the data itself. And so when you get down to the scale of individual axons, there's no information at all in the diffusion imaging of brains. And this is what the actual data look like. And this is one 1/1000th of a voxel in diffusion imaging. And you can see axons in the white matter of macaque, and you can see axons going in every single direction. And unlike the textbooks, the axons in the white matter of macaque and human brains, we have data as well, are rarely organized the way the textbook or caricature might give you on the left here, but often they have, you know, some structure that you can see, and with not fascicles, but groups of neurons going in different directions. But often on the right, they're going in almost random directions if you look at it. So how do we collect these data? It's actually quite simple. It's far easier than EM connectomics. It's just you can use one of many types of 3D light microscopy for the confocal, or better, a light sheet microscope. And you can stain all axons or all axons of the large axons with antibodies. And there's enough room between them, both in the gray and the white matter, that you can actually trace them all. Whether machine learning can do this at the large scale, time will tell.
Excuse me, 10 minutes.
10 minutes, oh good. I am in my not so many final slides. So what you saw so far are with antibody stained axons in the white matter collected with light microscopy. And you know, as with Google Earth, you know, this is you know, the goal is to create data sets, in fact, quite a bit larger than Google Earth. But the way to do this is with lots of very fast cameras. You know, how did we get speed with electron microscopy, with transmission EM and fast cameras? How can we get all the data that we need for this fluorescence axon tracing with fast cameras attached to light microscopes? And this is a schematic of a light sheet microscope. Keyword light sheet, look it up if you don't know what they are, but it's a wonderful form of very fast three-dimensional microscopy. Final data slide, but really just, again, examples, but showing what this looks like in a mouse. Rather, in this slide, rather than just showing the axons, which are labeled in green, I'm also showing the myelin in red and the cell nuclei in blue. So here's a cross-section through half of a mouse brain where you can see the very thin strip of white matter underneath the cortex here. Here you can see the basal ganglia and the striatum, where there are lots of bundles of axons and, you know, hippocampus and other brain areas, midbrain areas. Here is an example of the border between white matter and cortex in a mouse. Again, 3 colors. It's nice to see the nuclei. It's nice to see the myelin, to see different sizes of neurons, myelinated and even some unmyelinated. And you can trace these large axons quite nicely in the cortex, but even when they're tightly fasciculating, as they do in the mouse, you can trace them here in the white matter. Here's a fun example from the striatum here, showing one way of how we can get at different cell types. This is one fascicle within the striatum. And you can see the green labeled, which are the neurofilament labeling, and then the red circles with no center, where you can't even see the axon, but you can see the myelin, but not the axon. Different neurons have different proteins, and therefore different antibodies will trace them. And here's an example of being able to see 2 different classes of projection neurons according to their antibody tracing. So that's really it for data. It's very early days of this project, but we have all of the fundamentals in place. And very importantly, we have all of the big data handling, stitching, aligning, et cetera, pipelines that need modification, but that have been made robust from the electron microscopy. So where are we? The human white matter axonal connectome has the potential to provide a modern map of the cortical and subcortical areas defined by architecture and patterns of interconnectivity. And this is really one reason to give this talk. I really would love it if the modeling community started thinking about not just the subway map view of brain areas, but actually the point-to-point mapping between brain areas and what that means for computation. It'll have long-term clinical importance. I'll skip over this for this crowd. It'll certainly give a complete cell atlas for the human brain with antibodies for molecular labeling. And here's a very brief aside for machine learning. IARPA and local circuits, part of the inspiration for that was understanding the computation, underlying machine learning. You know, what is our computer for doing machine learning? And really, what is our computer, the local computational unit? But the axonal connectome will give a network of networks. And what is the overall architecture of the complex learning system that is the human brain? And certainly it'll be the largest biological network to date, and just, you know, and almost done. So the atlas of the human brain is a long-term goal, you know, experimentally and really because of the large data size. But when achieved, and whether this is 4 years, 7 years, 10 years from now, I think it will be achieved. It'll be 10 to the 10 axons, roughly 50,000, roughly at least half of the long distance axons, or at least the myelinated axons in the brain. Data sizes are huge, 1 exabyte, but intermediate goals are certainly the mouse brain at this scale, which I think will happen in the next year or two, and the macaque brain sort of geometrically in between those two extremes. We're not alone in our dreams, and the Brain Initiative has recently said that there are going to be 2 transformative projects, really huge projects that will be funded by the Brain Initiative. One of those is Connectomics, and it's really an either or both. They have 2 projects for Connectomics. One is the whole mouse brain connectome, which I could have talked about in the second half of the talk. And I'm more than a casual observer in this field. A lot of us are really pushing for it. But what I've spoken about is the second thing that the Brain Initiative is pushing as well: attain a whole primate brain projectome, first in the non-human primate, and then in the human. So finally, what are the big challenges in this field? Data collection and data handling. Going from where we are now, the petascale, to the exascale is a major jump. It's engineering, but it's a major jump. We have to make pipelines, and certainly the data processing, you know, going from the pictures. I've given this once before to a less neuroscience-y crowd. It's like going from Google Earth to Google Maps. The pictures to the maps themselves, we need segmentation. It's going to be expensive to do segmentation. And finally, data sharing. There are tools for looking at all these data, both in EM and for the projection mapping. But how can we share these data to maximize their use by modelers? And this, in the few minutes of discussion, we'll have not just connection matrices, but detailed high-resolution morphologies, synapse locations, and synapse sizes for the EM connectome. So here is the small but growing team for Axonal Connectomics. Really, all the data I showed were collected by an RA, Emily Tershak, and a collaborator at MIT in Janelia, Ray Gao. And this is the huge team of EM, and Nuna will go through this as well. And I'll stop there. Thanks.
Thank you very much, Clay. Excellent talk, very exciting. So we don't have much time left, but let's try to take one or two questions quickly. So question from Gaute, how accurately can one estimate synaptic strands from EM recordings?
It's a long discussion in the field. And there are a number of sort of calibration studies that look at EM features in tissues that have been studied biophysically. But in excitatory synapses, synapse size and postsynaptic density area, things like that, vary quite nicely with synapse strength. Different types of synapses between cell types all need to be calibrated for this, but relative strength of a given pathway, the world believes pretty well that these proxies are a good approximation. There's a huge grant put together by Kristen Harris that we're part of, just starting now to deal with this question in detail. 24 groups, so stay tuned.
Okay, thank you. Well, let's maybe take one last question here from Xia. Here. How different are the connectomes from mouse to mouse? Can you offer some ideas on how to analyze the connectome given the incredible complexity of the data set?
Yeah, I mean, I wish we had from mouse to mouse. We have the platinum mouse. We have, there's a grand total of 5 volumes that I've been involved with, maybe another 6 cortical volumes. We don't know, but as with anything biological, biological intuitions will tell you what things are idiosyncratic, like a fingerprint, and what things are likely to be quite robust from the literature where you are able to have a large N. And I think exactly what Nunu is going to talk about, cell type specific connectivity, from a lot of literature, that's likely to be fairly conserved or even quite conserved between species. So every neuron is unique. How can you classify? I don't know, but biology is full of rules, and I truly believe that like-to-like connectivity, neurons that do the same thing and that tend to fire together tend to wire together. And that's been proven many ways, including by electron microscopy and physiology. So, but the real answer is, this is just the beginning. Let's not do the Human Genome Project because it's one genome, but now we have thousands. So, to the future.
Okay, thank you, Clay. Thanks very much.